All right, let us continue chapter 2. And specifically, we left off talking about membrane-bound organelles. Let's talk about lysosomes. So, lysosomes are going to be pouches, essentially, that contain enzymes that will degrade cellular or extracellular debris. They are going to do this through one of two different processes, either endocytosis or phagocytosis. We're going to talk about those in more detail when we talk about transport mechanisms, but basically phagocytosis is going to be when the cell engulfs something, eats it, and endocytosis is going to be when it pouches it to brings it in. Again, we'll talk about that more with the transport processes. All right. Lysosomes can actually be divided up into different specific components. For instance, if we're talking about a peroxisome is a lysosome, but it contains a very specific compound. Specifically, peroxisomes are going to be used to degrade certain types of waste molecules, excess amino acids, certain types of fatty acids, and toxic foreign substances, because what they contain is an enzyme such as catalase. By breaking down these amino acids, fatty acids, and specific toxic foreign substances, they create a byproduct called H2O2. Some of you may already know this chemical formula. This is peroxide. Peroxide is deadly to your cells. It's one of the things we talk about in microbiology is how certain types of bacteria deal with this, especially our anaerobic bacteria, but peroxide is deadly. It is a poison. And so, compounds that are going to be broken down and give off peroxide as a byproduct are going to be broken down in these peroxisomes, because these peroxisomes are going to contain a special enzyme. This enzyme is known as catalase. And what makes catalase special? Catalase breaks down peroxide into water and oxygen, basically breaking it down into usable components. Our cells can take this water, can take this oxygen, and use it for a variety of different things, to be perfectly honest. So peroxisomes, super useful, and are going to be involved in that peroxide breakdown. Our non-membranous organelles, these are going to be organelles that do not have a membrane surrounding them. These are going to be things like ribosomes, vaults. We're really not going to talk about them either. They're a relatively new discovery. Again, though, they are mentioned in your book, so if you are interested, I strongly encourage you to read about them, but we are not going to focus on them in this class. And centrioles and the cytoskeleton. Ribosomes. I mentioned them in passing. What are they? Ribosomes are going to be a compound that are going to create our protein chains, our peptide chains. They are a combination of our ribosomal RNA and protein chains. They're super small, about 25 nanometers, and they can be either attached or fixed, which is attached to the rough ER, or free, meaning they are free floating through the cytoplasm, or cytosol, I should say, and they can make proteins out in the great beyond, so to speak. Super important, super useful. If a cell does not have ribosomes, it cannot make proteins. So, not all cells have ribosomes. Case in point are adipose tissue. So fat cells. They do not contain ribosomes. They really don't contain mitochondria either, because they don't need a lot of energy. But that's a different conversation. Centrioles, what are they? We're really not going to focus on them at all, except for right here. Centrioles are going to be paired cylindrical structures that sit perpendicular to each other, so think of a plus sign. And Unless the cell is actively undergoing mitosis, they're not doing anything. They're sitting there, chilling, hanging out. Because what they're going to be involved with is the mitotic spindle apparatus process, or part, I should say, of the mitotic process. So, for now, we're not worrying about them. We're actually not going to talk about mitosis in this classroom. You should have had mitosis in other biology-based courses. If you have not, I do encourage you to read those sections of the applicable chapters, but we're not going to focus on it. The cytoskeleton, this is a lattice of fibrous proteins that does a wide variety of things. They're going to give mechanical support and structure to the cell. They're going to aid in intracellular transport of goods within the cells. They're going to suspend the organelles and anchor them so they're not kind of all over the place. They're going to allow for the formation of adhesions with other cells. What's an adhesion? Think of it as a sticking point. It allows two cells to stick together. If the cell contracts, the cytoskeleton is going to be involved with that. If the cell moves, the cytoskeleton is going to be involved with that as well. If you're like, this is all over the place. It's all over 20 million different slides. How am I going to remember what slide contains what information? Don't worry. Your book and this PowerPoint contains a table summarizing this information. For instance, it has the cell part, whether or not it's a membrane-bound or non-membrane-bound, and its structure, so what it looks like, and then its function. So, Cliff Notes can be found on page 65. Alright, let's talk about those adhesions. Cell-to-cell adhesions, cell-to-cell sticking, cell-to-cell connection, however you want to phrase that. There's basically three types of these adhesions. And these are the three types that we're going to mention in passing in various systems, so you do want to be familiar with them because we're not always going to refresh our memory on them. One is going to be the tight junction, one is going to be the desmosome, and one is going to be a gap junction. What's a tight junction? Tight junctions, for the most part, are going to be typically found only in the epithelial tissues. And basically what tight junctions do is they limit molecular movement. Basically, we fuse two adjacent cells together, forming an impermeable barrier, or nearly impermeable, meaning they're fused together, there's no gaps, basically nothing can get past them. In order for something to get from one side to the other, it has to go through the cell. That's the goal of a tight junction. The big thing with tight junctions is, yes, they force things like sodium, potassium, etc. to go through the cell, but it also forces water to go through the cell. And that's the big part of it. These junctions help form watertight barriers. So blood plasma stays within the heart or stays within the blood vessels. Water stays outside of your cell or outside of your body or in your kidneys or in your bladder, wherever it may be. Your stomach acid stays in your stomach. Provided you have adequate mucous membranes as well, but because of these tight junctions. Watertight barrier. Desmosomes. Desmosomes are going to form more of a filamenty junction, and this is because we're not trying to lock them together. We want to have a little bit of give and take, because what desmosomes do is they bind cells together for strength, allowing them to stick together despite mechanical stress. So they don't tear apart and they don't let go or break in any way, shape, or form. So there's some give. Think of desmosomes as like spandex. Stretch. Gap junctions, on the other hand, are going to link the cytosol of two adjacent cells together. So they're going to allow for the easy movement of ions and other molecules to pass between the cells. Think of it as like channels. This is going to allow for direct communication between two adjacent cells. And this is going to be important with various systems.